Right, everyone, welcome. Thanks for joining us. So today's ERI seminar, we've got Dr. Graham McCullough from University of Otago joining us. So Graham did his undergrad and PhD down at Otago. He's postdoced at the University of Queensland. He's now back at the University of Otago working as a researcher. Been involved in a couple of Marsden projects. And yeah, I mean, the work he's going to talk to today is about New Zealand stream insects, particularly one of our stonefly species, Aotearoa, and some really interesting evolutionary patterns. So yeah, I'll let Graham take over. Thanks.
Sure. All right. Thank you very much, Frank. Thank you for organising us, and thank you, Joshua, for helping do this, and thank you, Chrissy, for the invite. She invited me a couple of years ago. So I'm only about 2 years too late. So today I'm going to talk about these guys here. These are stoneflies, and these guys are really cool. And if you're not convinced they're cool now, hopefully you will be by the end of the seminar. So as Frank said, I'm a researcher at University of Otago, and most of my research involves using genomic tools to understand really cool questions about evolution and ecology. So this is back in 2006. Believe it or not, that's me. There was no grey hair. The reason for that is it's before I did my PhD and also before I had kids. And so in 2006, I decided to do a PhD. And so I did it with John over there with the stonefly. And it's fair to say John knew absolutely nothing about stoneflies when we started. And I knew even less. And so now this is Team Stonefly in 2024. Frank, you feature in this. And so we have a number of different people involved in this now. So we've got a huge number of students involved who have driven a huge amount of this research. So a lot of this research is actually driven by students, and I'll try to point that out as we go. And then we've got collaborators. We've got physiologists. We've got people who work on stonefly behaviour. We've even got someone who's a psychologist in there, strangely. And so it's a really diverse group we've got now, and it's really exciting. So today I'm going to talk about anthropogenic evolution. So if people know about anthropogenic evolution, the cool thing about it is it happens really fast. So we're not looking at something that's going to happen over millions of years. We're looking at something that's happening over tens of years or hundreds of years. And probably the most iconic example of this is these moths here. So these are peppered moths. Back in about 1800, everyone in Britain was collecting peppered moths, probably because there was nothing else to do back then. And so every moth they found back then was this peppered colour. We then went through the Industrial Revolution, so late 1700s, early 1800s. And what happens is all the trees suddenly got dark. They got covered in smog. We can see in this bottom picture there. And suddenly it was actually an advantage to be melanic. So by about 1900, 98% of the moths were melanic. So that's pretty incredible, isn't it? Within 100 years, you've gone from almost completely peppered to almost completely melanic. So in the last 30, 40 years, there's been a real push to clean up all the smog. And suddenly the trees have changed colour again. So the trees are now back to what they were before. We can see in this top photo. And suddenly it's an advantage to be peppered again. And so the most recent survey in about 2010, about 90% of them were back to that peppered colour. And so this is a really iconic example of anthropogenic evolution. What I'm going to talk to you today is about two examples of anthropogenic evolution in New Zealand insects. These two examples are even better than these for two reasons. One is about some endemic species from Aotearoa, and two, because it's about stoneflies. And the main driver we're looking at here is deforestation. So that's when people cut down or burn down the forests. And here we have a picture of the South Island. Some of you North Islanders may not recognise it, but this is the South Island. This is the other half of New Zealand. And it's undergone extensive deforestation. So almost more than 70% of our forests have been lost since the arrival of humans. So initially there was a lot of burning when the Polynesians arrived, and that really intensified, particularly in the last couple of 100 years when Europeans arrived. And so now we have a huge amount of deforested areas. And so when you have this deforestation, you present the animals that are in the surviving area a couple of different options: disperse or die, or maybe evolve. And when you look at that picture there, it's actually quite a stark contrast, isn't it? You've got the nice green there, and then you've got a completely different habitat when it's deforested. Not only don't you have the trees, you have everything associated with that. You have different predators, you have different wind conditions, you have different humidity, different sun. And so it's quite a big change. And so here we are, back to the hero of the show, the stonefly. So if you don't know stoneflies, you will know them soon. So stoneflies are stream invertebrates. They have nymphs that live in the streams for between 1 to 4 years, depending on the species. Then they have these very short-lived adults. And so the adults are living for maybe 1 to 4 weeks. And then they'll fly around, they'll mate back into the stream and go back into nymphs again. And so the first aspect I'm going to be looking at is wings. So we're looking at anthropogenic evolution of stonefly flight. And so, as I said, nymphs turn into these adults, but in some cases, particularly in New Zealand, we end up with these guys. So these here are wingless adults. This is a stonefly that our team discovered in Maungatua, which is just overlooking Dunedin Airport about 5 years ago. So that black stonefly, as you can see, it's got absolutely no wings. And about a quarter of our stoneflies have no wings at all. And that's really unique. So we don't have that anywhere else in the world. And this is actually what appealed to me initially when I started to work on the system, was just the huge number of species that seem to have no wings. I wanted to know why they didn't have wings and what was the genetic basis for it. Why does this keep happening? And as you can see, they actually look really cool. So there's 3 examples of some stoneflies. And all that we knew when I started my PhD was they're up in alpine environments. And so most of my last 15 years, I spent a lot of time going up to these alpine environments. So I've got a couple of students there. I've got Kahu up there and Anne around there, Brody on the other picture. And we spend a lot of time going up to these environments, collecting these samples, which you can imagine is actually a really fun thing to do. And one of the first things I was really interested in is it the alpine environment? So that was what was recorded as causing this wing loss, but there wasn't really any evidence for it. And so this here is a picture, a figure showing full wing species. On our y-axis, we've got elevation. Don't worry about the x-axis at the moment. The full wing species are in black, and the wingless species are in red. And so when we look at it like this, it seems like elevation seems to be a pretty good proxy for whether they have wings or not. It's worth noting that this is actually combined across a whole lot of different species. So, for example, if we said 1,000 meters as our cutoff, that sort of differentiates most of the full wing species from the vestigial wing species or the short wing species. You can move it up to there, still looks like pretty good. What we really want to do is put a line through there though. So that line there would separate everything above that line would be red, so it would be vestigial winged or short winged, and most of below that line would be black. And that line is actually a real line. So on the x-axis, we actually have the elevation of the local tree line. So we can see a picture there of the tree line. So this is when you go up a mountain, this is as far up the mountain as the trees go. And so what this is saying that anything above that line is above the tree line, and almost always it's got short wings. Below that line, It's almost always got full wings. And so what this is saying, it's not elevation per se that's causing these things to lose their wings. It's something about being above that tree line. And the really cool thing is we can actually look at this also within a species. So this is the Landoperla fenestrata. This is the first stonefly that I ever really worked on. And it's actually got two different morphs. We've got a big full-winged morph, you can see there. And it's also got one there with vestigial wings, or really tiny little wings. And both of these morphs are found basically throughout the Lower South Island. So this map here shows them, the vestigial wing ones in red and the full wing ones in blue. So you can see they're all over the place. And so we can do the same sort of analysis with these guys. It's not quite as clean as when we look at those interspecific comparisons. But again, you can see that almost always above the tree line, things have short wings. Below the tree line, they have full wings. And we do have a couple of places there where we find both the full winged and the vestigial wing ones together. And one of the really cool things we found is you can actually see this transition when you go walk up a stream. So these are two streams on the Rock and Pillar Range, which is about an hour inland from Dunedin. And what we did is we walked up these streams and took samples every 100 metres. And then we could record whether they were short-winged or full-winged. And this graph here shows as graphing elevation versus the proportion of vestigial winged morphs. So at the bottom, at about 200, 300 metres, everything's got full winged. As we started making our way up the mountain, at about 600 metres, we started to notice, oh, there's some vestigial wing ones coming in. And eventually, when you got to the top of the mountain, everything was vestigial winged. So you can actually see this transition within individual streams. So these are two individual streams, about 5 k's apart, and we're seeing the same transition in both streams. And in both cases, this transition seems to be associated with the tree line. So basically below the tree line, everything's full winged. The moment you get to the tree line, you're sort of starting to get a 50-50 mix. And then by the time you're 100 metres above that tree line, everything's short winged again. So what's actually causing this? Why is this tree line so important? Well, the reason we think is quite obvious when you're actually out there, it's the wind. So when you're doing the sampling, you're in the trees, it's nice and calm, there's no wind around, everything seems fine. The moment you get out of those trees, the wind picks up. And particularly up at those, on the Rock and Pillars, the moment you get out of those trees, it is really windy. And so what we think is, when they're above the tree line, these things can't really survive up there without wings. They're not good flyers to start with, and they're stream invertebrates, so they very much have to get back to their stream. The moment you get blown away from your stream, you're gone from the population. So you've got two options there. You either get bigger and better wings, and a lot of insects do that, or as the stoneflies do, they just give up and get rid of their wings. And so this is actually really important. What we know now is that it's the forest that seems to be linked to wing loss rather than altitude. And so then when we come back and think about deforestation, we can actually start to make predictions what's going to happen when you cut down forests. So we have a potential conceptual idea of what's going to happen there. So a long time ago, everything was forested. We would expect most things to be full winged. As those forests get cut down, we would expect obviously less forest cover, so less that are going to be full winged. And then in areas where there's no forest, we expect those wings to completely disappear. A lot of this work was driven by Brodie there. We can see he was a PhD student of ours, currently in Brisbane. And what he was particularly interested in is the tree lines. And so we've got two pictures here. One is of a natural tree line. You can see it's quite impressive. It's basically a straight line. That's at about 1,100 metres. But what actually happens when you have a lot of burning is your tree lines actually get lowered. So it burns everything at the top of the tree line, everything at the top of the gullies, and all you end up with is a little bit of remnant tree in the gully. And this tree line can be 200, 300, 400 metres lower than it was originally. And so we looked at 5 locations here. And what we did is we did the same sort of transects that we did on the rock and pillars in these different locations. So to see what sort of transition we get from full winged ones to reduced winged ones. The only one with a natural tree line is Borlandburn. So this is in Fiordland. It's got a natural tree line at about 1,100 meters. And then we had 4 locations with lowered tree lines. So these tree lines had been artificially lowered because of burning. And so our first figure there is showing the frequency of flightless ones versus the elevation. You can see they all show these curves where you start off with full winged ones at the bottom and they go almost fixed to vestigial winged ones at the top. But you can see this occurs at different elevations. We can then correct that to where the tree line is. And what you can see is almost in every single location, it's all to do with our alpine tree line. So if you're below that tree line, you're full winged. If you're above the tree line, you're vestigial winged. But the crucial thing is here is these tree lines have actually moved. These tree lines weren't actually always at this location. So this is where these tree lines used to be. They used to be 100, 200, 300 meters further upstream. So what we've actually seen has happened is we've actually seen this really rapid adaptation. So these populations used to be full winged, and then the tree line's gone, and then they've suddenly become short winged, which is really cool. And so we got some really cool publicity for this. This is about 4 or 5 years ago. That was in the New York Times when insect lost their homes, evolution clipped their wings. And then this has just come out in New Zealand Geographic. Check it out if you get a chance. By Kate Evans and Giselle Clarkson, just talking about this wing loss. And so we know that deforestation has lowered these tree lines and it's caused this sort of rapid evolution. But what actually happens in the lowland areas? So we know that there's huge swathes of the Otago region that are completely now deforested. So these would have all been, even at lowlands, would have been completely covered in forests. And so this is led by Gracie, a master's student of ours who finished last year. And what we did is we looked for a sort of similar to what we had with those elevational gradients. This time we actually had a latitudinal gradient. So at the bottom at Jimmy's Creek, site number 1, that's completely forested. Beaumont and Canadian have a bit of forest, a bit of scrub. The Willows has a bit of scrub. And then eventually we go to places which have completely lost their forest. And so do we see that same transition in the lowland as we see going up a mountain? And the answer is yes. So the pie charts here show the proportion of full winged ones in black and your vestigial winged ones or reduced winged ones in white. So the 4 to the south, completely full of full winged ones. As we start to transition into this deforested area, we start to get mixtures. And then when we're fully deforested, we've completely lost wings again. And again, this stuff has only been around for 800 years, this lack of forest. So 800 years ago, this would have been fully forested, everything would have been fully winged. So we see this transition happening really fast. It's just another way to look at it, looking at these sort of particular clines, showing they show the same sort of pattern as we see when we go up the elevational clines. So that's really exciting. What I'm particularly interested in, as I said at the start, is the genomics. There was a couple of big questions I was interested in. One, are the full wing ones and vestigial wing ones related? How are they related to each other? Are they 2 different gene pools or are they the same? I'd done some mitochondrial markers, which suggested there was no difference. But what will we find with genomic markers? And the second thing is to find the gene that's involved, or the genes that are involved in this. And so what we did is we sampled full wing ones and vestigial wing ones from 8 populations. So these 8 populations have where we know we have mixtures of both forms. We then did some genotyping by sequencing. So that's getting the genome, cutting it all up and comparing sequences across them. So we've got about 50,000 SNPs across the genome. So it's sort of giving us this genome-wide analysis of what's going on. The first thing to notice here is we ended up with 3 different groups. So we got a fiordland group, a rock and pillar group, and an umbrellas mountain group. And I'll have a look in a bit more detail on these groups. This is fiordland. This is up Mount Burns track, which is an amazing track if anyone gets a chance to go up there. And what we were expecting to find is one of 2 things. Either we were going to find everything is mixed together, so there's no difference between the full wing ones and the vestigial wing ones, or we were expecting to find maybe all the full wing ones would be in one group and all the vestigial wing ones would be in a different group. We found something completely different. So this is a PCA showing the relationships among the different populations. And in fact, we actually found 3 groups. So all the full wing ones together, so at the bottom of the streams, they were all together. But what we found was there was actually 2 completely independent vestigial wing populations. So what that means is these 2 populations at the top are different from the populations at the bottom, and they're also different from each other, which is really unexpected. This suggests that they've actually probably gone up to the top of that mountain and been isolated for quite a long time. And we found exactly the same pattern on the rock and pillar range. 3 different groups. So the full wing ones were all together, and then the 2 vestigial wing populations, again, different from each other. And this is really incredible because these populations, in this case, are only probably 3 k's apart, and they're just completely different. And then this is our central group. And here we found a different pattern, completely different pattern again. So these ones are all reasonably closely clustered together, but we can separate them out by stream. But in this case, we found no difference between the full winged ones and the vestigial winged ones within a stream. So why is that? Why are we getting this different pattern in different regions? So most of these places, the tree line is at about, or the mountain itself is probably only goes up to 1,000 meters. So 800 years ago, before there's been any deforestation, there would have been probably no vestigial wing ones up there at all. Everything on that mountain would have been full winged. So only in the last 800 years, when we've had this little bit of deforestation, has there been this habitat for these vestigial wing ones to come. So again, what we've got here is we've probably got new wing loss in each of these locations. So if you cut down the trees, suddenly these things start losing their wings. And so the question I always had is, why is there so much wing loss? How come every time you go to the top of a mountain, you find these vestigial wing ones? It's a really interesting thing when you think about it, because if you lose your wings, you don't actually spread around much, which makes it quite hard to explain. And how come every time we have this deforestation, these things seem to rapidly adapt and lose their wings? And so there's probably a couple of different options from a genomic perspective anyway. So one, we've got different de novo mutations at each location. So each time they're coming up with a different solution to their conditions and losing their wings. The second option is you have a mutation once, and then that spreads to all the different places. And so we wanted to know which of those 2 it was. And so we've been looking at this sort of trying to find the genes involved in wing loss for at least the last 7 years. What I'm going to talk to you about is the most recent one, and these are results we've only got through in the last couple of weeks. And what we did here is we went to places where we know we had mixtures of them. We had mixtures of full-winged ones and short-winged ones. We took 40 of each, and we did whole genome resequencing. And so that's trying to get the whole genomes of each of these individuals and compare them across each other. We then look for outliers. And by outliers, I mean particular SNPs in the genome that differentiate the full-winged ones and the vestigial winged ones. And so for each of those locations, we had outliers. So we had outliers at 6 Mile, Blackjacks, and Mount Burns. And then we wanted to look for ones that were shared across all of those locations. And we found one gene with a whole lot of outliers in it across each of those locations. And so we got really excited. We thought, right, we found the gene. This is excellent. It's going to be one of the genes that they've worked on in Drosophila, because they know about 10 genes in Drosophila or more than that are involved in wing loss. They all have really easy names to remember, like vestigial, wingless, apterous. And then we've got the gene name, and it's angiogenic factor with G-patch and FHA domains. So it has no known role at this stage in wing loss. We know angiogenic means it's got something to do with blood vessel development, but that's all we really know about it. So what we then did is we went, well, we know it seems to be involved in these 3 locations. How about we look at other locations where we have this sort of mixture? And so we went to, I think there's about 12 or 13 locations there where we had a mixture. We took a few full winged and a full reduced wing ones, and we sequenced the first exon in that gene. And because it's a coding sequence, we could then translate that to amino acids. When we looked at it initially, we didn't see anything that interesting when we looked at it, just looking at the bases. And so here we can see we've got a whole lot of samples here. I haven't shown them all on here. So example at the top, it says full winged N equals 10. I've only shown 5, but we've actually got 10 in there. And then we've got the full winged N equals 30. Probably only got half of them in there. If we start looking at the full winged ones and vestigial winged ones from Fiordland, what we found is a 5 amino acid deletion in there that differentiated the full wing ones and the vestigial wing ones. And that happened at every single sample. And so we're reasonably confident that this is what's causing the difference between the full wing ones and the vestigial wing ones. When we looked at the central Otago ones, didn't seem to have that deletion, but we found one amino acid change that was consistent across every single one. So that's across 30 vestigial wing ones and 30 full wing ones. If you got an A there, you got full wings. And if you got an S there, you got vestigial wings. So A for all your wing, S for short wing. And so that was really exciting. So it seems to suggest we have these shared mutations underpinning wing loss. But in different regions, they've sort of come up with these different solutions. In Fiordland, we have one mutation. In central Otago, we seem to have a different mutation. And probably the way I think about this is evolution at 2 time frames. So you've got this ancient evolution. It's maybe not ancient, it might have happened over 50,000, 100,000 years ago, or maybe a bit older than that, where this vestigial winged ecotype has evolved up in the mountains and spread around. It's become really advantageous. And then we have this contemporary evolution. So deforestation, we see these guys turning up again. But in this case, they're not reinventing the wheel. They're not coming up with new solutions. They're using this sort of mutation that was developed a long time ago for the alpine conditions, and it's now becoming really useful in these lowland deforested areas. And so that's the wings. What I'm going to talk about now is colour. I'm going to give a little disclaimer here. Please do not share this. This has just been embargoed by quite a major scientific journal. So enjoy this. Forget about it for 2 weeks, and when it comes out, just go nuts. So again, this is something that was led by Brodie Foster. And so again, we're looking at exactly the same species of stonefly. As well as losing wings, it has 2 different colour morphs. So we've got a brown colour morph, and we've got one which we call a melanic colour morph. So the question is, why do we have these 2 colour morphs? So also in the environment, we have this guy here. This is Ostroperla. And when you look at it, you can see it looks almost identical to Zelandoperla fenestrata. And so I've got a picture of the 2 on the fern there. In the field, you can't really tell them apart. You can tell Ostroperla has slightly squarer, slightly shorter tails, but really they look very similar. And so one thing you can also notice, as well as being black, they have some very distinctive colour patterns. So on the forearm, they've got a yellow, and on the shoulder, they've got a really white patch too. I get in trouble with entomologists by saying that insects have forearms and shoulder patches. And so I keep doing it to wind them up mainly. And so you guys will probably actually recognize what this color pattern is. You see it a lot in the insects, and you even see it in some frogs and some other vertebrates. And so this is warning coloration. And what we know about Aotearoa is it's actually toxic. And so it's full of cyanide, it feeds on wood. And if you squish it, it smells like almonds. Don't squish native insects, people. And so what we think is happening is that these Aotearoa fenestrata are actually mimicking the Aotearoa. And what's actually incredible is this color pattern is even seen in some vertebrates and some mammals. So warning coloration says stay away. Some people don't listen. Sorry, I had to quickly transition from that slide. I actually looked for a picture of Chris Luxon with the yellow tie, but he didn't have one. David Seymour did have one, but I was a bit slow to put it on there. And so we were interested in the stonefly color. And so what we did is we took photographs of each of these, and then from that, we looked at particular parts of it, and we derived a melanization index. So this is showing how melanic these things are. So looking at that graph on the -4 scale, not very melanic. Further to the other side, about 3, 4, 5, they're very melanic. And then we can actually do exactly the same thing with Aotearoa, and we can see where they overlap. And anything that overlapped, we could consider maybe a mimic. Anything that doesn't overlap, non-mimic. And so we were quite excited about that. But the question we kept getting is it really mimicry? And what people were really trying to say are the birds actually avoiding the melanic Aotearoa? Our normal response was, of course it's mimicry, just go out and look. But apparently reviewers don't accept that as an answer. And so we actually had to demonstrate this. So our first thing we did was some predation experiments. So these are stoneflies. They have been dead for several years. They were stored in a freezer. We got them out, pinned them to a tree, and our plan was to see whether birds eat them. So we put a non-mimic and a mimic beside each other, and hopefully the birds were going to avoid the mimics. We'd put these out, walked away for 20 minutes, came back, wasps were absolutely all over them. Several of them have been decapitated already, and so this wasn't going to work. We went and talked to a behavioural entomologist in our department. She was very excited that the wasps were eating them. In fact, she asked we were quite distraught at this stage. She asked for details about how they ate them, which part of the brain they ate. But luckily, she actually came up with a solution for us. And that was clay models. And so this is quite often used in behavioural ecology. And so what we did is we made clay models of these things. We made them identical, apart from one was made with brown clay and one was made with black clay. And then we set them up, as you can see on the other photo, beside each other in the streams, see what would eat them. And so, yeah, 50 pairs and then a couple of deforested sites. And the good thing about clay is you don't need to set up cameras because you can actually see whether the birds or other insects have eaten them by the marks they leave. So at the top, we can see the sort of typical marks you'd get from a bird, quite direct, quite often triangular. Rodents have very distinctive marks based on their teeth. And sometimes you'd find these little pitted marks, which are normally insects. And so focusing mainly on the bird predation, because that's what we're interested in the forests, about 25% of them have been attacked within, I think we set them up for 3 days. Almost, a quarter of them have been attacked in the forest of the non-melanic ones. And then the melanic ones, less than 5%. So that's a significant difference. And so this seems to suggest that birds are going for the brown ones and not going for the melanic ones, which really supports our argument. We also set them up in deforested areas. As you'd expect, significantly less attack rate because there's not that many birds around. And as far as we could tell, the birds weren't really discriminating. So the birds probably didn't know that the melanic ones were poisonous there, which is great. In fact, there was actually more attack on the melanic ones than the non-melanic ones outside the forest, probably because they stood out more. And so hopefully that's convinced you it actually is mimicry. What again I was interested in is the genetic basis of mimicry. So what's causing the mimicry? As I said, with the wing loss stuff, this can be quite hard. You spend many years on it. Our first thing we tried was genotyping by sequencing approach. So cutting up the genome, comparing these regions across, I think it was 48 melanic and 48 non-melanic ones. What we've got here is an outlier scan. All those black dots, there's about 200,000 of them, are showing regions of the genome that don't differ between melanic and non-melanic. And then we've got 6 regions that do differ. The one region that really stood out is a gene called ebony. As you can see here, this is known in Drosophila, and it makes Drosophila mutations in it make Drosophila black. So we managed to find that gene really easily. So I think the moral of the story there is either you need to try for 10 years, just keep trying, or you just need a bit of luck. As luck would have it, there was also a 7 base pair indel right beside this SNP. And this indel seemed to differentiate the melanic ones and the non-melanic ones. And this is really useful for us because collecting stonefly nymphs is much easier than collecting adults. So you can go to the stream and you can go get 100, 200 nymphs quite easily. To get that many adults would take you 10, 20 trips back to that stream. And in fact, what we normally do is we get them as nymphs and then raise them in the lab. And so now we had this marker that would actually tell us whether things would be melanic or not. So this is again looking at the melanism index. So that same melanism index we looked at before. But in this case, we've sorted them out by genotype. So everything with the low allele was non-melanic. Everything that was heterozygous was almost certainly non-melanic. And everything with that high allele, almost everything, was melanic. And so this seems to explain over 70% of the colour variation. What I was particularly interested in when Brodie was doing this was the distribution of this mimicry. And so he always noted that mimicry only seemed to be found at certain sites. And so when he was looking for example, a population where he wanted to get 48 of each, he said, well, you've got to go to the Leith, you've got to go to Mongatua to get it because it's the only place where there's lots of mimicry. So he looked at samples from across the lower South Island. Anything that had a mimic, you can see is covered and is marked with yellow. Anything without mimicry is marked with blue. So you can see there's a lot more places with blue than there are with mimics. And what we didn't actually notice is it seems to be associated with forests, which kind of makes sense when we think about it. So we know that Ostroperla, the thing they're mimicking, feeds on wood. That's how it makes its cyanide. And it's particularly common in forests. But then you go out of the forest, it's quite rare. And so we would expect to see exactly the same pattern with our Zelandoperla mimics. We would expect them maybe to be common in the forest. Then the moment you get out of the forest, they're going to be a bit more rare. Rare for a couple of reasons. One, they stand out. So that bright banding is actually making them stand out. And if birds don't think they're poisonous, they're just standing out for no reason and getting eaten. And also we know that melanic pigment is actually quite expensive to produce. So if you're going to produce all this melanism and there's no benefit to it, it's just a waste of resources. And so this particular research was led by Stephen Nee there. This was actually his honours project. And as we can start making a prediction, like we did with the wings, what's going to happen when we get rid of the forest? So let's say 800 years ago, lots of melanic ones. As that forest disappears, the proportion of melanic ones decreases. And then when the forest's gone, the proportion of melanic ones completely disappears. Okay, so we did some sampling in different streams. Again, lower South Island, this is the best place to be sampling. We did them from nice forested streams. If you look at that map, there's not that much forest, but it's actually sampling from forested streams is very easy because whenever there's a nice bit of forest, DOC kindly puts a nice track right through the forest, straight to the streams. Sampling from deforested streams, on the other hand, is actually a bit harder. There's a lot of deforested area, but it's all been converted into farmland. And for some reason, farmers don't really like you scroffling around in their streams. And so again, the first thing we did was some genotyping by sequencing. This is to look at the relationships among the different samples, and then among the melanic ones and the non-melanic ones in each region. So what we found is we actually had 4 subclades sorted by region. And the important thing here is also to note is we have mimics and non-mimics within each subclade. And looking at this, there's no real difference between the mimics and the non-mimics genetically. And so we can actually have to start to have a look at these streams. And so these are 2 streams. They are 4 kilometres apart. At Whiskey, we found over 50% of them were melanic. And then we go to a deforested stony stream, 20% of them were melanic. So quite a big drop in melanism in that deforested area. And here's another pair. They're very close together. Chloris, again, only a couple of kilometres apart from Gilfillan. At Chloris, about 40% of them are melanic. At Gilfillan, 14%. So almost a third decrease again. And then we can look at that across the whole of the Lower South Island. So on that side, we've got the deforested ones. The key with that is looking at the brown, almost everything is not melanic. You go into the forest, 40% seem to be melanic. Some of you will notice that at the top there, samples F1 and D1 seem to have a lot of melanism, including D1, which is, it's actually obscured there in the thing, but it's just below that no tweeting sign. Seems to be almost fixed for mimicry. And so these are 2 samples at the northern end of the range. So they're just north of the Waitaki River. And when we look at them genetically, we actually find these are completely fixed for melanism. And this is actually a really important point. So what's happened is we assume sometime in the past, these things have lost that brown allele. They've become fixed for melanism. And now in the deforested area, they're still fixed for melanism. There's no genetic material for them to work on. So they can't actually shift the same way as these other ones do. And then Stephen put this through some fancy stats, taking into account latitude, longitude, the relationship among populations. And this is basically looking at the mimicry frequency across these different populations. So in our deforested one, we've got a mimicry frequency of typically about 10%. We go to the forested ones, always a lot higher, 30, 40, 50%. And then we've got a summary overall, which suggests, again, 3-fold reduction in the deforested zones. What you will notice is that even in the forest, in most cases, that frequency sort of hovers somewhere between 30% and 50%. It doesn't seem to get fixed. And what we think is going on there is some frequency dependent selection. So if that was at 100% and these particular stoneflies were significantly outnumbering the Osteoperla, when a bird ate it, most of the time when a bird ate one of these stoneflies, it wouldn't actually have any problems because it would be eating these guys here, which aren't toxic. And so the key is you don't actually want to be outnumbering the thing that you're mimicking. You don't want to be outnumbering the toxic model. So that's why we think it's not typically not at 100%. This is again looking at it with that genetic marker, exactly the same thing as we found last time. And it seems to be that same mutation linked to melanism across all populations. And again, explaining about 70% of the variation in colour. And what we can actually look at, because we actually have some samples from different known timeframes of deforestation, we can actually look at the rate of melanism loss with deforestation. And so on that x-axis, we have deforestation timing. So this is the number of years before present when this deforestation happened. And then on that y-axis, we have the frequency of this ebony mutant. So in green here, these are our currently forested sites. So again, at 40 to 70% frequency of this mimicry, of this melanism allele. We know some of these sites were deforested during the initial burning period, so Polynesian burning. And then we also have some sites around, for example, the Otago Peninsula, which were deforested a bit more recently. So maybe only 100, 150 years ago. And from that, we can actually start to estimate how quickly deforestation is causing this shift in mimicry. And so we've got 3 lines there. These are from our 3 different subclades that we mentioned. And what we can see is there's very similar trends across all these different subclades. So no matter where you are, if you lose the forest, you lose mimicry at a reasonably consistent rate. This is about 1 to 2% per generation. And so what was really exciting about this system is it's actually quite a complex system involving a whole lot of species interactions. So that first thing I talked about with the wings, we have quite a simple thing. You lose forest, suddenly it gets windy, you lose your wings. This one, we've actually got a whole lot of things involved. We've got predators, we've got a model, and then we've got the predators eating the model, and we've got a whole lot going on. And so this deforestation seems to completely break up all these particular relationships, and which then causes this melanism to disappear. And so the reason this is embargoed, this has just been accepted into science about 3 days ago. So we're obviously quite excited about it. What I've done here is I actually highlighted those ones in bold. Stephen, Gracie, Brody, and Kahu are all students, which is really exciting. In the case of Stephen, this was his honours project. Kahu was doing a summer project with us at the time. And this is his first paper, so hopefully he just keeps going up from here. And so I'll just quickly finish up looking at potential future shifts. And so what I think was really cool about the moth example that I mentioned to start with is that we had a shift to melanic moths, and then as the pollution disappeared, we had a shift back in the other direction. And so the question is, will we see that same shift with these stoneflies? And this is quite a big question now, because we've got a huge amount of forestry happening. Both we've got planting of plantations, and then we've had this real push over the last 10 years to really reforest with native forest. And so this is work led by Kahu, who's doing a honours project at the moment. And if we are to be believed, the government has funded 627,000, I don't know, million, 627 million trees to be planted over the last 8 years. And so this is some of the sampling Kahu and I did at the start of the year. What we've got is 2 photographs at 2 different dates. One is Robert Creek back in 1985. We did not do sampling back in 1985. Kahu wasn't born until about 20 years after that. What we did is we just sampled in Robert Creek in 2022. This is just south of Queenstown. The point of these 2 photos is at the top, you can see almost everything is deforested, apart from a little bit of native forest at the side. And then now there's been these plantations. And so there's been, in the last 30, 40 years, we've seen this change from deforested habitat to this plantation. And so then we looked at those 2 aspects we looked at. We looked at melanism, has that shifted? And then we looked at wings. So we looked at the frequency of the ebony allele in the native forest. It was about 40%. We then looked at the deforested area, and obviously that's going to be lower, and it was higher. Okay, so no shift between the native forest and the deforested area. Why is that? We're not quite sure. We're looking at quite a small distance between the forest and the deforested area, probably only 1 kilometre. And perhaps the birds are actually flying across that area. Perhaps there's gene flow across the stoneflies across that area. So we're not actually seeing that drop that we've seen in other regions. Plantation doesn't change either. So let's try wings. Maybe we have some more lake. In the native forest, every single thing was winged, which is what we expect in native forests. The forested area, that's better, down to 25%. So when you go into that deforested zone, most things have lost their wings. Then we look at the reforested area, starting to shoot back up again. So what it looks like is when you put this plantation in, these things seem to be gaining their wings. And this is only, we're looking at probably 30 years. We've gone from 25% up to 70%. And so this is very preliminary at this stage, but it's something we're going to keep exploring as we go and will be the focus over probably the next couple of years of research. That's all I've got for now. So just thank you to University of Otago for paying my salary, and again to Marsden Fund for paying my salary. A few acknowledgements there. And again, thank you to Chrissy for inviting me, Frank for introducing me, Joshua for sorting everything out. Thank you.
So I think we've got time for questions. Question, Ange?
Hi, great talk, thank you. Just wondering, for that future part that you finished on, do you have any access to temporal samples, like museum samples that could complement that work?
Unfortunately, no. So people don't tend to collect, well, they collect stoneflies, but not in the numbers that we need. So for a lot of these things, you need 50, 100 samples to actually get them. We've had one person in the South Island, Brian Patrick, who's collected samples, but generally it's only been 2 or 3 samples per site. So yeah, that would be ideal though.
Space for time design and at a broader spatial scale and set for that, but yeah, still looks pretty good with the wingless forms. Other questions? Joshua?
While you were collecting the other information, were there any patterns associated with the generalized count and size differences of individual stoneflies between the forested and deforested areas?
Not something we've looked at specifically. Generally there is a size difference between the full-winged ones and the vestigial winged ones. Sometimes the vestigial winged ones are much smaller and sometimes they're much larger. So whatever that means, I don't know.
Yeah, that could be driven by site conditions of temperatures and things like that, so it's probably independent. But yeah, Christy, you got a question?
Yeah. So you're talking about, obviously, with your wingless and winged, it's a genetic polymorphism. Of course, one of the other ways that animals can kind of adapt to new situations is through phenotypic plasticity. Is there any examples of wing loss? Is it always a genetic polymorphism, or is it also a plastic conditional response?
It could very well be plastic in some situations. So what I'm focusing on here in particular is the difference between the full wing ones and the vestigial wing ones. We also have another form of wing loss, which is quite common, where they're only not wing loss, it's just wing reduction. And so it might only be a 20% wing reduction up in alpine conditions. And I suspect that may be more plastic. It doesn't seem to be associated with the markers that we've found so far, but yeah, potentially.
Charlie, you got a question?
The reviewers may address this, but I'm very curious about those clay mimicry experiments, because birds are tetrachromats and we're trichromats. Is there a long history of using clay as a way to replicate?
Yes, the answer is yes. Yeah, luckily there seems to be a lot of information, a lot of papers published in that. The paint we used was a UV paint that they always seem to use. And so the birds, they see that UV and apparently it's very bright for them. So yeah, they seemed happy with it. It's a good question though.
But on that note, there's a lot of papers where they've used caterpillar mimics them and done predation experiments that have been published in Science as well. Now I've got a question. I've got a few questions actually, so we'll go after lunch. But in the sub-Antarctic islands, I think there's a few stoneflies that are wingless. I'm just wondering if you guys have had any thoughts about investigating that, because of course that would be the adaptation towards windy conditions. But it'd be interesting to think about the evolutionary history there with when they colonized and then obviously lost in their wings.
Yeah, so there is, Auckland Islands is about 5 species, and the good thing there is I think there's 3 wingless ones and 2 that are winged. I suspect that one of the winged ones and the wingless ones are the same species. And so these sort of studies have a lot more power when you've actually got these polymorphic species. We've done some work on them. All we know is that they colonized those islands 5-10 million years ago and haven't had a chance to go down there and have a look at them, but they would be quite cool.
Yeah, no, cool. Angela, question?
Just thinking about the obvious effects of wingless status on gene flow. Are you also interested in looking at the interaction between like wingless or winglessness and other traits like the melanism or some other traits?
Yep. Yeah, definitely looking at those things. And so, yeah, it's kind of hard to work out how these things are moving around if they're losing their wings. We suspect it's probably an ancestral polymorphism, so something that's been in the population for a number of years and then is spreading through. Initially, we thought through winged heterozygotes, but our last lot of sequencing, every single heterozygous stonefly happened to be wingless, which suggests it may actually be a dominant mutation, which makes it harder to explain how it's spreading around. But yeah, we'll definitely explore those sort of things in future.
I think you might have something on the chat too.
Someone on Zoom got a question. Sorry.
So if you just kind of hover over the where it says, just kind of hover over where it says you are screen sharing.
Yeah. Oh,
Okay.
Oh, just saying.
No sound yet.
Well, good.
Cool.
Yeah, one last question. I just, the mimicry system's just cool, but so complex. I think about geographical mosaic theory of co-evolution and how there's different moving parts there. With the clay models, did you get any sense of the type of birds that were doing the predation? Just because I think about, obviously our avifauna has taken such a hit and such an impact of invasive, non-native mammals, where, that adds another sort of, complicating factor into the system.
Yeah, so we don't know at this stage. We're going to set up some cameras over summer. We suspect it's probably Piwakawaka as one of the major ones, because every time we went to the streams to put these clay models down, they would come down, they would be interested in what we're doing. Possibly something like New Zealand robins in some places might be another good option.
Yeah.
Yeah.
Okay, yeah, cool. All right. No other questions? I think we'll give another hand of applause for publication in science as well. It's awesome. So, thanks a lot, everyone.